So the last time we left off about how we, this really great system that bacteria have to deal with viral infections, right? And to prevent their genome from being infected by viruses. And that was the system we talked about with the CRISPR Cas9 system. We discussed briefly how we, as scientists, has leveraged this system to alter genes to study their function in the genome. And the way that we're able to do that is taking advantage of two key features, and that is this Cas9 protein, which is required for cleaving the target that this guide RNA will then recognizes. Okay, so you need a Cas9 and you need a guide RNA that's designed by you to find the location within the genome. So the guide RNA is usually designed based on where you want to target in the genome. Okay. So this complex enables us to find any region in the genome where we want to alter or modify in any way. This also requires that adjacent to that guide RNA, there's a photospacer adjacent motif, or a PAM site, it's often referred to. This is important because the Cas9 protein recognizes this site, and that's where it makes that double-stranded break upstream of this site. Okay, so not only do you need to take the Cas9 protein to the region that you want with the guide RNA, it needs to be adjacent to this PAM site. And that's what really makes this system really specific, right? Because you're really targeting a particular region and not just random DNA in the genome. This PAM site is found only in the invading DNA, so it's not present in bacteria of its own, right? Or in the system, in any eukaryotic system, right? It's something that it's not present in bacteria. In eukaryotes, we look, when we design the guide RNA, we look for this region because that's what we are trying to apply this CRISPR-Cas9 system to use. Okay, so we need to find a site within the genome that contains this PAM site so that we can target it to that location. For bacteria, this PAM site is important because it really distinguishes its own DNA from the invading DNA. So this is not present in bacteria, so you know that Cas9 will only cleave that invading viral DNA and not the bacterial DNA. And the way that this CRISPR is able to modify genomes is it creates double-strand breaks, right? So when you have this, in this case, you have this complex, right, which is your Cas9 and your guide RNA. This guide RNA recognizes a region in the genome, right? So this is a double-stranded DNA in the genome. And it also recognizes this PAM site, right? Because it's also there. Once it attaches and binds to that region, it creates a double-strand break, okay? You guys recall, with double-strand breaks in the genome, they can be repaired in either of two ways, right? It can be repaired by a non-homologous end joining or homology-directed repair, right? In the non-homologous end joining, this situation allows you to create either an insertion or a deletion, right? Because it's not going to be repaired perfectly, right? You have DNase polymerase 1 that's going to chew back some of this DNA and add bases to fill it in, right? So you can either end up with sequences that were deleted or sequences that gets inserted randomly. And that's what creates the mutation, because you're modifying this DNA. If it's a gene, you're modifying that gene and adding or deleting bases. So you're really modifying the DNA. If homologous, you can also use this system in the case of homologous directed repair, where you have regions where you know what that region is. You provide in that reaction a donor DNA with something that you want to insert into the genome. You put this into the genome along with this Cas9 guide RNA complex. when this cleavage happens, you have this donor DNA that will recombine because the ends of this donor DNA is similar, and you will have recombination, homology-based recombination that will help to insert that new DNA. So again, you're inserting a new piece of DNA, a new gene, you're modifying the genome. Okay. There's other ways that you can use, and this is really the minimal amount of modification ways you can use Cas9, right? You can also use this Cas9 to just cleave one strand, right? So there's a modified version of Cas9, which is called Cas9 nakase, where it can only recognize and bind to a single strand of DNA, right? So you can either do, one strand here or one strand here, target multiple regions within the genome this way, again, and add a piece of DNA that you're interested in into that region. You can also use Cas9 to look where in the genome you want to, for instance, use an activator or repressor. So let's say, for instance, you want to regulate a particular gene that's nearby. You can design a guide RNA for a region adjacent to that gene that you want to regulate, attach to this Cas9 guide RNA complex an activator protein, so that when it gets to this location, this activator protein can then act on that gene that you want to regulate. You can do the same thing with a repressor protein. You can fuse a repressor domain to this Cas9 protein guide RNA complex, and you know where it's going. You know where the gene is. It's adjacent to the gene. It can serve as a repressor. Again, this is a way to really precisely regulate a gene within a genome in vivo, right, in the organism. You can also look at localization. So you can check to see where within the cell your gene is located, right? So you can use it for visualization. So again, to this Cas9 guide RNA complex, you add a fluorescent protein, right? The green fluorescent protein, which will fluoresce green under a microscope, and you can visually look where within the cell that protein is located, okay? So it's really a beautiful system to do many different types of modifications. We have now, this is another way, we have used it to manipulate all levels of gene regulation across many different organisms, right? And when I mean many different levels of gene regulation, starting with DNA replication, starting with, transcription, translation, protein abundance, accumulation. We can do many different modifications that we want. We can add DNA to the genome that wasn't there before, again, using these different Cas9 guide RNA complex, depending on the Cas9 you use, right? So I told you that bacteria have many different Cas proteins. Cas9 is the one that's most commonly used. There's a number of them that have been modified since to be used, such as Cas12 or Cas13. The dCas9 you might see, and that's the reason that you use a dCas9, it means a deactivated Cas9. You would use a deactivated Cas9 for cases where you have this activator repressor, as I mentioned, because you don't want to cleave the DNA. So you need to deactivate the Cas9 because you don't want it to cleave the DNA. You just want your Cas9 and your guide RNA to go to this region and bring in that activator repressor. So then you need to deactivate this Cas9 so it doesn't cleave it, right? So these are different versions that you can make. You can directly edit RNA, right? And you can also target the ribosome, yeah? So you can also regulate the level of translation as well. There's a really great video that I encourage you guys to watch on YouTube, which really precisely explain how this complex is going to the RNA, to the DNA in the genome with that guide RNA and Cas9. The cleavage has happened, it shows you the repair and the outcome. It's really beautiful. I think you should try to definitely look at that if you're able to, okay? So now we know that currently, right, this is from 2014, but it still holds through. it's being used in every area of biological research, regardless of what the organism is, to manipulate many different processes within the cell, right? We can alter genes, you can alter DNA, you can target whatever you want to target that particular location. This discovery, of course, clearly, so yeah, the link should be here you should be able to copy this link. It should be present in the lecture slides when I learn, so you should be able to just copy this link and find it. If you don't, let me know. Yeah. So, and for Hussam, it doesn't cut at the restriction side. It really cuts randomly, right? So, oops. So this PAM site is here. This Cas9 protein cuts 6 to 9 base pairs upstream of this PAM site. So it's it's varied, right? There's no restriction enzyme site. It just cleaves there. That's the nature of this enzymatic activity, right?
I thought it had to be like palindromic site because of the name of CRISPR, because it stands for clustered, regularly interspaced palindromic site or short palindromic site. So I thought it had to be palindromic or something.
No, it's not. No. So the name from CRISPR comes because of the way that it's in the bacterial genome and how those get clustered in that spacer region, right, as the sequences comes in. And because of the order of how those clusters are on both strands of the DNA, right, that's palindromic, right? So it's like both of those strands, but that's in bacteria. It's not necessarily the way that the Cas9 protein works. That's just that space, that's just the region within the bacterial genome that that takes a version of, that integrates the DNA, the invading viral DNA. And that's where the name comes from that region.
Oh, okay.
Yeah. So, but the Cas9 protein of itself is not a restriction in the nucleus in that context where it recognizes, a restriction site to cleave. We just know that it cleaves 6 to 8 base pairs upstream of this PAM site. Yeah. And that's probably the way that the enzyme wraps around, recognizes the DNA and how it sits on the DNA, that region, that 6 to 9 base pair region is where it actually cleaves. Yeah. So yeah, so of course this was a major discovery for everyone. And in 2020, the Nobel Prize went to the 2 women who worked extensively on the system. So they won the Nobel Prize in chemistry. Jennifer Doudna, who was at UC Berkeley, so one of the UC researchers, along with her colleague in Berlin, helped to identify this system and how it works. It's a beautiful system. At a time when they were working on it, they did not anticipate that it was going to be such a major breakthrough, right? I mean, Jennifer Doudna, of course, did not expect to win the Nobel Prize for it. She was very shocked and surprised, right? Because again, it's all they're using this system. They're chemists. They're trying to understand how a certain process works, and they were not necessarily the pioneers, because the understanding of CRISPR and the way that bacteria deal with invading viruses, research has been going on for decades, right? So a lot of people were working on this. I mean, they've seen this type of CRISPR system before, but no one really understood what was happening, So they really took advantage of previous research from many different groups and, just found a different way to look at it. And the people working in our lab, you can hear, if you ever have a chance to listen to Jennifer give a talk, I mean, they were just doing research like everyone else working in the lab. And just trying to get to a certain level of understanding, and did not expect that their discovery was going to just completely revolutionize the field, right, of biological research. So it's really amazing. So it's always like you really don't know what your discoveries, how your discoveries are going to be used, and what impact it will have on society. So it was really a really nice story from these 2 scientists in the discovery of CRISPR. So it's being used everywhere. So that's kind of the last thing I want to really talk about. I think as you're studying with CRISPR, of course, you want to make sure that you understand the differences between RNAi and the CRISPR technology, the types for RNAi, the types of rules of small non-coding RNA in gene regulation, right? We talked about 3 main types. The proteins and nucleus is involved and what they do. Understanding the mechanisms of CRISPR and the important components, right? We talked about having this Cas9 protein. We talked about having this guide RNA. We talked about in terms of the application for different organisms. We talked about you have to have this PAM site, right? So if you're given a DNA sequence and you need to determine where your guide RNA will bind, you need to take into account not just where the guide RNA shares homology within that DNA, but is there a PAM site nearby? And the feature of a PAM site is NGG. That N can be any nucleotide. It can be AGG, TGG, CGG, or GGG. Really try to understand that, okay, in terms of designing this type of experiment. Similarities and differences between CRISPR and RNAi, and what are the ways in which CRISPR can be used to alter genomes, right? And I think coming back to particularly this slide would really help you in terms of understanding what are some of the ways, okay? If you have to describe 2 ways in which you can alter genomes, right? You know that you can create mutations, and you also know that you can activate or repress a gene. You can also look at localization, right? So think about it in that context, in things that you can do with CRISPR technology. And if you get a chance, look at this video because it really summarizes it very nicely with beautiful videos and graphics to check that out. And yeah, so that's what I would think about. So before we go in, since your mind is already completely thinking about RNAi from the last class, and I like to give you these questions where I know that you didn't just see it, to see how much of the information you were able to compare with the new lecture. So RNAi is stimulated in the presence of what in the cell? Okay, that's in that. So, okay, so it's also kind of mixed, right? But 63% of you mentioned that it's double-stranded RNA, and it is double-stranded RNA, right? So you have the double-stranded RNA that's either from a virus with an RNA genome, whether it's from microRNAs that forms these double-stranded RNA from the precursor RNA, could be from transposons, right, forming this double-stranded, but that is one of the key differences between RNAi and CRISPR, is that RNAi, you're looking at it's a double-stranded RNA molecule that really triggers this RNAi pathway compared to in the CRISPR system where it's a double-stranded DNA molecule, right, that triggers this, okay? So go through, try to understand the differences between these 2 pathways. There's going to be several questions on comparing these mechanisms. Okay, so let's talk about, let's switch gears and talk about translation. So very beginning of the class, we talked about DNA replication, right, in both eukaryotes and bacteria. We also talked about transcription, right, in both organisms. And what happens once the RNA is made. We didn't go into too much detail about RNA replication, right? I mean, you can make more and more of RNA. So this is kind of, if you think about the way that you kind of sustain that post-transcriptional or the RNAi pathway, right? You're replicating the RNA and making more of it. Now we're switch gears and talk about what happens at translation, right? So once you have the RNA, you've already processed the RNA, you've done your 5' capping, polyadenylation, it's been modified in many different ways. You have a stable messenger RNA, which somehow amazingly got out into the cytoplasm to be translated, okay? And that's what translation is. Right, is a process by which the ribosome reads that final genetic message in the mRNA that produces a protein product according to the message. And that message in the mRNA is really coming from the DNA, right? Your initial DNA, that's where everything comes from, okay? And in order for translation to work efficiently, you need to have a number of important players, right, that helps with this translation process. So the ribosomes are super important, right? They're protein factories. They need to be able to acquire the amino acids, the transfer RNA, that's important as well, to translate that RNA into a peptide or a protein, okay? And join together the different amino acids that matches whatever that RNA codon is into a polypeptide. And this is it's a complicated process, right? Because, and you will see that both eukaryote and prokaryotes, while most of this information is shared, there are some differences, right? So this is kind of a overview of what translation looks like, right? So you have these big gray bubbles, which are your ribosomes. The amino acids are in green. And you have your tRNA that's shown in these, I don't know, pink, orange kind of color. tRNAs contain the amino acid, right? So they will bring in the amino acid. The tRNA knows which base pairs in your mRNA it needs to bind to based on regions within that tRNA, which are the anticodon sites, right? So they usually match the RNA. And they get recruited with this ribosome to add these amino acids that are coming in, and these amino acid kind of gets added along the way, and you have your growing polypeptide or your protein that's being made, okay? You know, this is mRNA, 5' to 3', right? The ribosome is going along from reading the message from 5' to 3' with the coding information and just adding the bases and the amino acid. The growing polypeptide chain also you will see have directionality. So the first one is the N terminus, and the last one that gets added is the C terminus for the amino acid. How does the ribosome know which amino acid to add to to match with your mRNA? You know that there are different codons in the amino acid that are composed of your 4 bases, right? It's RNA, so it's U instead of T, right? You have this genetic code that is made up of, it's a triplet code, so it's 3 different nucleotides, okay? So every 3 bases represents a particular codon. This code is degenerate, right? So that means that you can have more than 1 codon per amino acid. So for example, let's say we're looking at leucine here. Leucine can be, you know, CUU, right? CUG, CUC, many different codons will code for the same leucine amino acid. So that's why the code is considered to be degenerate. We have 3 stop codons, which doesn't really code for amino acid. It really just tells you, okay, this is the end. This is where you need to stop making that polypeptide. So if you were to consider that you have 3 different three different types of, the triple code is 3 different bases, and you have 4 different amino acids. You have 64 possible codons. But from these 64 possible codons that you can have, 3 of them are stop codons. So it's really 61 that's really coding for these 20 naturally occurring amino acids, right? For that particular protein. And this is the basis of the genetic code. Okay, it's 3 base pairs, a triplet code. It is degenerate, so, and it also have 3 stop codons that tells you that you should terminate making that protein at this particular location. And that's with the codon. In addition to the codon, you have the rest of the translation machinery, right? We talked about the ribosomes. The ribosomes are really composed of proteins and ribosomal RNA. Okay, remember the spliceosomal, remember when you had your spliceosomal complex, you had your spliceosomal proteins, and you also had these short nuclear RNAs that's part of this spliceosomal complex. It's the same with the ribosomes, right? You have protein and you have a ribosomal RNA that comes along with this complex. They usually are 2 different size in terms of the subunits, and they usually separate to attach to the mRNA. When translation is ready to proceed, they come back together. We'll talk about that in a minute. And in addition, you have these transfer RNAs. These are the small classes of the small RNAs that will read the genetic code, bring the correct amino acid. And they will bring the correct amino acid based on regions in this tRNA that recognizes those 3 bases or that particular codon in the mRNA. What's different between prokaryotes and eukaryotes? So in prokaryotes, you know that everything kind of happens all at the same time, right? And in the same compartment. So there's no cytoplasm to nucleus for prokaryotes in bacteria. So as your mRNA is made, it immediately starts being translated, and this polypeptide chain is added. However, in eukaryotes, it's a little bit different, right? You have your nucleus. All of what we talked about in the last 2 or 3 lectures is involving what's happening here, right? Your mRNA is being made, you're modifying your pre-mRNA to mature messenger RNA, where you have the 5' cap, polyadenylation site, it's already spliced out. That gets transported into the cytoplasm, and then it gets translated, right? So it's really compartmentalized in eukaryotes. Besides these 2 processes, you'll see that most of translation is very similar in terms of the steps in translation and the proteins that's involved in translation, even though you will expect to have more in eukaryotes because it's a more complex system. So the ribosomes is also a little bit different between eukaryotes and prokaryotes, right? So depending on the final ribosomal protein composition, you can have different types of ribosomal RNA and different types of proteins, right? So in the case of prokaryotes, and that's what the pro stands for, you have the 2 subunits, large and small subunit. Each of these subunits will have their own type of RNA, right? And they also will have different composition of proteins as well for different subunits. The sizes are going to be different. They are usually referred to as, whatever the number is, S, and this S is really just Sedberg's unit, and it's kind of a sedimentation unit. So these unit is just based on how fast the ribosome hits the bottom of a tube when you use a centrifuge to spin it out, right? So it's kind of a sedimentation, how quickly it gets to the bottom. So, and also the size of each of these subunits is not proportional to the mass. So that's why 50 plus 30 doesn't equal 80, right? When they combine together and you spin them down, this is the estimated the speed that they will spin down to, okay? Eukaryotes is very similar to, you also have ribosomal RNAs that are specific to the large or small subunit and different proteins that are involved in this process as well. If you were to look at that within those complexes, right, you have your ribosomal RNA, you have these triangles and colors, and circles just mean different types of proteins for these subunits. They assemble as a form, right? So they assemble in this context where you have the protein and the ribosomal RNA together in one of these subunits. So this is the large subunit and this is the small subunit. And within those subunits are ribosomal RNA and proteins, okay? The ribosomal RNA, it's RNA. The structure is completely complex, right? So there's lots and lots of secondary structures, right? And you see all of these kind of looking like hairpin structures because this RNA very easily folds back on itself, right? So there's lots of secondary structure for these ribosomal RNAs, which is okay because you really just need one region of this ribosomal RNA, as you'll see, to bind with your messenger RNA, okay, to bring that subunit to that base. Like I mentioned, it's base pairs with the mRNA. It recognizes regions within your messenger RNA. And these ribosomal RNAs also have peptidyl transferase activity. This activity is important for adding the polypeptides together. Okay, and you'll see that a little bit later. So they help to bind to the mRNA, and they also have this peptidyl transferase activity for these ribosomal RNAs. So the way that these tRNAs, right, and ribosomal RNAs are usually made, right, they're not transcribed by RNA polymerase 2, right? They're either RNA polymerase 1 or RNA polymerase 3, depending on which one it is. In eukaryotes, right? In bacteria, they're usually transcribed because bacteria only have one RNA polymerase. The way that these tRNAs and RNAs are transcribed in bacteria, it's a large transcript that is then later processed into smaller parts, okay? Compared to in eukaryotes, you have separate tRNAs and separate RNAs, genes that exist and say, okay, you need to transcribe this particular RNA, okay? RNA polymerase 3 in eukaryotes are particularly responsible for the tRNAs and the ribosomal RNA. But there are some that RNA pol1 will transcribe, but most of them are transcribed by RNA polymerase 3. The other thing that's important for this mature tRNA is that at the end of the mature tRNA, there are these modified bases and a CCA at the 3' end of that base. You'll see why that's important in a minute. In bacteria, this bacterial tRNA, the CCA is part of the gene. Okay, so the gene gets made, the CCA is encoded, it gets added. Some other bacterial tRNAs and all of the eukaryote tRNAs, the CCA gets added at the end. So it gets added as modified bases to the end of the tRNA, okay? And it's not dependent on any kind of template. So there's a polymerase that will add the CCA to that end of that tRNA in eukaryotes, okay? The other thing that's interesting to know is that some eukaryotic tRNAs genes also contain introns, right? We talked a lot about introns, and introns are present in messenger RNA. We know that some tRNAs also contain introns that needs to be spliced out before they can be properly used. And this do happen as well with a different group of spliceosomal proteins that recognizes and splices it out. There's 3 stages of translation. All 3 of these stages are shared across eukaryotes. Don't. Okay, we're still there, right? Okay, you guys can still see the slides, right?
Yeah.
Okay, sorry, Leah. So there's 3 phases of translation. And in the initiation phase, and this is all shared between bacteria and eukaryotes, right? You have an initiation phase. The ribosome need to bind to the mRNA. The first amino acid gets added. And that comes in because it's attached to the tRNA, which also binds that complex. You have an elongation phase, right? So the ribosome is adding the amino acids one at a time. And then you have a termination phase where that polypeptide gets released because the ribosome has encountered one of those 3 different stop codons that we talked about. In order for translation initiation to occur, there are 2 important events that must happen before this can take place, okay? One of them is you need to have a supply of amino acyl tRNAs, right? These are the tRNAs that have the amino acid bound to these tRNAs, okay? This process of adding the amino acid to the tRNA, right? Because it doesn't just have, it's not encoded in the tRNA, right? The amino acid gets added to the tRNA. And this process of addition is this addition of this amino acid to the tRNA is referred to as tRNA charging. So you're charging the tRNA because you're adding this amino acid to it. And the process that you're doing this is referred to as charging. And we'll see what I mean in a minute. The second thing that needs to happen is that you need to disassociate the ribosomes into 2 subunits, right? Because the complex of the small and large subunits together cannot assemble directly on the messenger RNA with the tRNA unless they're separate, okay? So that needs to happen. And the cell usually assembles this entire initiation complex on the small ribosomal subunit, okay? So they need to separate in order to make this small ribosomal subunit accessible. So these are the 2 main things that need to happen before initiation of translation can occur, okay? The way that this tRNA molecule gets charged, so this is an image of what a tRNA molecule looks like. It has really 4 regions. We're not going to talk about these 2 regions. What's important is this anticodon region, because this is the region that will recognize the bases, the codons in your messenger RNA. Okay, so it will be complementary to whatever this anticodon is here. Also at the 3' end, right, of that tRNA, I told you that the CCA gets added, okay, to this adenosine that's present right here. So it's added right there, okay. And as you say, the terminal adenosine is a target for charging that amino acid. So sorry, this adenosine here is the place for charging that amino acid. So that amino acid comes in, binds to this adenine that's present right here. The way that this happens is it really takes advantage of the structure of the tRNA. So what happens is that you have this amino acid that gets attached. So this is the amino acid in pink. It gets attached to either the 2' OH or the 3' OH of this tRNA with the adenosine at the end. Depending on, and it's really not known what the preference is, whether it's the 2' OH or the 3' OH. What we do know is that regardless of whether it gets added at a 2' OH or the 3' OH, at the end of the before it can get incorporated into translation, this amino acid, sorry, this is a 2', 3', this amino acid needs to get added or incorporated into the 3' OH. So if it gets added at 2' OH, it gets shifted to that 3' OH before you can actually start the translation process. So the way that the charging works is really because it's absorbing energy. So you have your amino acid. This amino acid gets a phosphate group from ATP, right? You know, with ATP, it's an energy source in the cell. Cleavage of a phosphate bond really generates a lot of energy. And so ATP, with the help of a particular enzyme called an amino acyl tRNA synthetase, adds this phosphate group to this amino acid, right? So it adds a single phosphate and monophosphate. So you have this AMP with these phosphate groups. The other two phosphate goes away, okay? So this addition of this amino acid to AMP charges it because it's an energy source. You're breaking the bond, you have a lot of energy. This energy gets transferred to this amino acid so that the amino acid is now charged, okay? Once this amino acid is, and I should go through this. So once amino acid is charged, then it combines with the tRNA, yeah, and this amino acid can then get added to this tRNA again, either the 3' or 2' OH of this, this tRNA that's present, right? So you combine them with that energy. Again, you have amino acyl tRNA, that's important. And then you lose your AMP, it goes away. Okay, and now you have a completely charged molecule, including the amino acid bound to your tRNA that you need. And now you can begin to initiate translation. What is also important to know is that these amino acyl tRNA synthase, there are 20 for each amino acid. So they're highly specific. Okay, so the amino acyl tRNA syntase that adds, let's say, leucine to this tRNA is specific to leucine. Okay, so they're highly specific. If there is a mistake, you incorporate the wrong amino acid, and you can imagine if you're making a protein with the wrong amino acid, you will really affect the function of that protein. So they're highly specific. Of course, mistakes can be made, and it does actually happen, but you need to be very specific. Yes, Christian.
You were saying that that the mistakes do happen, everything, if something's taken to the wrong spot. So what is there as a step to like help prevent that? Because I know along like making the mRNA and everything and through the whole process, there's so many checks. So what happens if it ends up like instead of adding a leucine, it adds an arginine?
Yeah, so that protein will become non-functional because you'll see, there's really no major proofreading mechanism for that. I mean, you just have to hope that the next round when this mRNA gets translated, it gets corrected. But that protein, depending on the protein that it's supposed to be, will get degraded, because you will see after when we talk about the proteins, once a protein is made, there is another quality check, meaning that they need to be folded properly, and they also need to go to the right location for their work. In either of those two steps, it's kind of a quality check. Yeah, so if there's a mistake in your protein and it's and if that mistake changes the way that your protein would fold, or changes the signal that sends it to the right, that needs to go to the right place, it gets destroyed and degraded in the system. Yeah, so that's kind of where it can be checked. Yeah, or your protein just becomes non-functional, even if it bypasses all of those checks. Yeah.
So then even if it bypasses all the checks, it's just non-functional and just degrades itself. Okay.
Yeah, it just doesn't do what it's supposed to do. Yeah.
Okay, thank you.
Mm-hmm. So you have your amino acid that's charged with the tRNA, it's attached to it. The next step we mentioned is you need to dissociate the ribosomes, right? You need to separate that large subunit from the small subunit. And once this happens, then you can start building that initiation complex. In both bacteria and eukaryotes, right, it's on that small subunit. So you have, after that dissociation, you have your 30S ribosomal subunit, which is small. In eukaryotes, it's 40S. You have your mRNA, right, which you're going to be translating. You have your amino acyl tRNA. This is the tRNA charged with your amino acid. And then you have a range of other proteins called initiation factors, as you'll see, which are important. Together, what's highlighted in red is known as the 30S initiation complex. Okay, and this complex also exists for eukaryotes, right? It's just you will have a different composition of initiation factors, and instead of the 30S, it will be the 40S. Okay. And the steps, again, this look like if you look at this model, it really looks like like it's a lot of steps, but it really isn't a lot of steps, right? Because we kind of discussed most of what we just discussed a lot of what these steps are, right? First, you dissociate your 70S, right, into 50S and 30S. This happens with the help of two types of proteins, right? The ribosome release factor and also an elongation factor. They help to separate these two subunits. Once they separate, you need to make sure that they remain separated, right? Because you want to build your initiation complex on a small subunit. So you don't want them to be able to easily come back together. The way that this happens is with the help of an initiation factor 3, which binds to this 30S and prevents this dissociation, right? Because it prevents this from binding here. You then have two other initiation factors, initiation factor 1 and initiation factor 2, that comes in and binds on this 30S small subunit. Initiation factor 2 have a GTP added to it because it helps to provide energy for this process to work. So it's usually bound to initiation factor 2. And it also helps with the release of all of these factors from this small subunit. So they all get bound there. When that happens, you then have the binding of your mRNA, right? This is what gets translated. And also your amino acyl tRNA, right? With the two together. So they all assemble on this small subunit. This is your 30S initiation complex. Messenger RNA, amino acid, amino acyl tRNA with amino acid, your 3 different initiation factors, okay, on this 30S subunit. Bacteria, you only have 3 initiation factors, so it's pretty easy, 1, 2, and 3. Again, just knowing what each of them do is important. And once this is assembled, then the large subunit can come in, but it comes in when release factor 1 and 3 gets removed. This is now free to assemble here. So this initiation factor 2, in a way, kind of recruits the binding of the 50S with the loss of these two. This initiation factor 2 will dissociate, right, with the help of GTP hydrolysis. But it's also kind of a way, there's also an hypothesis that this IF2 helps to recruit this 50S subunit to this complex, and then it gets released, okay? So once this is finally finished, you have this product of the 70S initiation complex that's ready to elongate. So it's very basic step. It dissociates, you prevent the dissociation with IF3, you assemble the entire initiation complex together, you remove these initiation factors and bring the subunits back together to begin translation. Okay. So usually the first codon that gets added is very specific. So in bacteria, there's 3 possible initiation codons, right? AUG, which codes for methionine, GUG, or UUG. Most times it's AUG, it can be GUG, or it can, in very rare cases, I would say maybe 1 to 3% of the time, you might have UUG. Okay, but primarily it's AUG, and that's what codes for methionine. In addition to this methionine being added to the tRNA, it's kind of a modified version of methionine. So added to this methionine is this formal group. So the initiating codon in bacteria is referred to as FMET, or N-formal methionine, right? This is the first amino acid that gets incorporated. This is different from another methionine that might get added in that polypeptide chain that's not at the beginning, right? It will just be a methionine. Okay, but the first one that gets added is a methionine that has a formal group. And somehow this is important for starting this initiation process in bacteria. This amino acid gets removed. In most times it gets removed from the protein during maturation. So it's not necessarily needed for protein maturation and protein function, but for translation, this is absolutely required. This was discovered through a series of experiments where they were trying to understand what is the composition of these amino acyl tRNAs. And what they found is that when they digested different amino acids, in this case, this is leucine, example of leucine, when they isolated the ribosomal complex, which is already bound to the mRNA to be translated, when they added RNAs, right, that will chew up the messenger RNA. And you can isolate what else is in that composition. So you have your nucleotides that gets cleaved by the RNAs, the ribosomal RNA. And what is left is your amino acid. So what they found was that they can see the structure of all these different amino acids, but when they did this with methionine, what they found was that the initiating methionine particularly was not the regular methionine. Instead, you had two versions. So they got the nucleotides, they got the methionine that's present within that polypeptide, and then they also had another type of methionine that had this formal group added to it. And this is what they realized, because it's usually just one of them, right? When you will have multiple other methionine, that this must be the initiating methionine within that cell. Okay, so that's how they discovered that this is what's happening in bacteria. So how does bacteria know, they have many, as I said, many methionine AUG codons within the RNA, right? How do they know where to start, right? How do you know where you would start? What do you guys think? Like, how does a ribosome know where to start translation? Where would that information be provided? Right, yeah, specific information on the RNA, right? So a while ago, many years ago, John Shine and Lynn Dalgarno were doing studies. Their work, of course, they work in translation in bacteria, trying to look to see what features in the messenger RNA signals the binding of that ribosome to begin. Okay, where does the ribosomal complex with the ribosomal RNA and the different proteins in the ribosomal complex, where do they recognize on that messenger RNA? Okay, and what they discovered was that there are sequences upstream of this a particular AUG, yeah, which seems to be shared between multiple different bacterial species, okay? And it wasn't just random, right? So what they noticed is that you had a purine-rich sequence, usually approximately 10 base pairs upstream of this start codon, right? This is not random, right? I mean, if you look at another AUG in the genome, you're not gonna find this, right? So they were able to look at the AUGs and see what is shared in those upstream of those sequences that determine that this is the place you need to start. And so they found this purine-rich region that's present here, and this is what's referred to as the Shine-Dalgarno sequence. This sequence is the sequence that interacts with the ribosomal RNA, right? Because remember, you're having ribosomal RNA coming in, that's part of that 30S subunit, right? So this is what recognizes that. And it's really present to start translation in bacteria. We'll talk about eukaryotes, how that's different in a minute. And they had to show and prove, right, using many different methods, that this ribosome is actually interacting particularly with that conserved sequence. And in order for that to happen, I'll get to your question in a minute, Hugo. And in order for this to happen, they had to do an experiment. What type of experiment do you think that they would have done to show that this ribosome is actually interacting with that conserved sequence in that messenger RNA? The ribosome, it's a ribosome, it's a ribosome, so it's a ribosomal complex. So you can think of it as a protein complex. It's interacting with nucleic acids. In this case, it's interacting with messenger RNA. What kind of experiment would you think you would do at this time? So with a filter binding assay, because this filter binding assay requires double-stranded DNA rather than a single-stranded molecule, a filter binding assay wouldn't necessarily work here. Exactly. So an RNA's footprint and type of assay, right? So we call it a nuclease protection type of assay, right? It's kind of the same idea, right? And that's exactly what they did, right? So they isolated this the RNA. They digested it with a nuclease, right? So the ribosome is bound to that site. You digest it. All of what's not protected will be cleaved. And then you have this sequence. You can then sequence and determine the sequence. And that's exactly what they did for a range of different complexes. And then you have all of these, you have your start codon, and then they can sequence everything and they can see that, okay, this sequence is usually about 10 base pairs upstream and it's conserved. In terms of why, the other question was, why does E. coli like high have the start as a GUG? Again, it just really varies. There really is no explanation why. It could be that perhaps, and this is all speculation that I'm giving you here, it could be that because the LAC-I operon is so highly regulated, that you probably don't want it to be as efficient the translation. I have no idea, but it's just that's just the way it is. Yeah, the LAC-I operon. That's the operon that codes for the repressor protein, right? So it might just be something related to the way that operon needs to be regulated. Yeah. So once you have the binding of the 30S initiation complex is formed, right? You then need to have that ribosomal RNA recognizing that Shine-Dalgarno sequence upstream of that initiation codon and binding to it. The region of the ribosomal RNA, and in this case, it will be the 16S ribosomal RNA that's in this 30S small subunit. It's usually the 3' end, right? So this is your ribosomal RNA. This is your messenger RNA that's here, right? These are different messenger RNAs for different genes. And you can see that you have this 3' region that will bind to this Shine-Dalgarno sequence, okay? And that's what actually binds and recognizes that sequence. Of course, this is related to the efficiency of translation, right? So if you have 100% homology, right? You can see here you have more homology, or here you have way more homology than here, and then here's less homology. So this would be really highly efficient at translation compared to one that's less, right? So it just depends on how that base pairing occur and how similar they are to this consensus sequence for the Shine-Dalgarno region, okay? So that is what happens in bacteria, right? In eukaryotes, it's a little bit different. So it also begins with a methionine, right? The methionine that gets incorporated first is not a N-formal methionine, but it also is a different methionine that's different from the methionine that gets incorporated later, and it's really called the initiation methionine, okay? There's no Shine-Delgarno sequence, as you would find in bacteria. But what helps with this start of translation is the 5' capping. Because remember, we talked about the 5' capping and poly A tail, where we say it helps with translation, right? You know, in addition to protecting the mRNA from being degraded, right? And that's what this 5' capping is absolutely important for many eukaryotic genes for translation. The way that this works is that you have your messenger RNA. This is eukaryotes now we're talking about. You have that 5' cap, right? With just a guanine that's been modified with a methyl group. The 40S subunit is a small subunit for eukaryotes. You also have these group of initiation factors. You have your aminoacyl tRNA, and you also have GTP, which is also required for this process. This small ribosomal subunit gets recruited by recognizing this cap. Once it gets to that mRNA, it scans along the mRNA looking for the start codon, okay? Once it reaches the start codon, then everything gets assembled, okay? This is not also random because you have many AUGs. Why does the ribosome stop at this AUG versus maybe another AUG that might be upstream or another AUG that might be downstream? Even though you don't have that Shine-Delgarno sequence, there are sequences that have been shown to be favorable, right? This was also done doing a series of mutations, doing a series of comparisons between different eukaryotic genes. And what they found was that there are certain rules for this AUG. And these rules are referred to as Kozak rules after the scientists who discovered it in the 70s, right? And these rules are just really a set of requirements, okay? So what they found to help to favor translation or the start of translation, that first AUG, is that you have your first AUG, and downstream of this AUG, they expect to have a guanine at this +4 position. So this is +1, 2, 3, 4. And also at the -3 position, a purine. Okay, so +1, -1, -2, -3. So you have this purine at this position. These are the rules. This AUG in this context is what seems to be preferred. Okay, and these are referred to as the Kozak rules. 5 to 10% of the time, most ribosomes will bypass that first AUG and look for one that's more favorable because these rules are not met. Yeah. The reason that we are able to determine that this 40S subunit really recognizes that AUG as the start codon, it's really with the help of that initiator tRNA, okay? So there's experiment, this is now a yeast assay, which you guys we haven't discussed a whole lot about. So these are experiments performed in a yeast. It's a eukaryote and it's an easy assay to do. And in yeast encode many different amino acids, in this case, histidine. If you create a mutation in histidine, or if you truncate the protein that makes histidine, then your yeast cells is not going to be able to grow on a media that doesn't contain histidine, okay? So you want to be able to produce histidine in your yeast cells because the media doesn't have it, and if you don't do that, then your cells will die, okay? So, and this is what they did. So what they did is that they created a tRNA where the anticodon was changed to UCC. It was changed to UCC because they wanted to have, instead of AUG, an AGG as the start codon, okay? And they did these experiments specifically to show that the tRNA really recognizes that codon in the messenger RNA, okay? So they converted this initiator tRNA codon to UCC, and in the histidine of itself, the initiation codon of the histidine, they had 5 different versions. And what you're seeing here, yeah, is if this tRNA binds to this initiation codon, if it binds, let's look at the bottom one first, if it binds at the right amino acid, you get translation. So the methionine is added, you get translation of this histidine gene in the right orientation. You can produce histidine, you have cells that's growing. In this context, what happens is that this anticodon somehow binds before this favorable one, right? So minus 28 base pairs upstream. Because it's not the correct one, it's also in the wrong frame, right? So the message is not read in the correct frame. And we didn't talk about the different frames, right? That you can, a protein can have 6 different frames, right? We didn't talk about those different frames that you can have. But it will be read in a different frame. And what happens is that, yes, you can make this protein, but somewhere it terminates prematurely, right? And the reason it terminates prematurely is because there probably is a stop codon here that shouldn't be here in this different frame that's being made. And you get termination, so the cells cannot grow because the protein is truncated, right? And this is really the way that they showed that, you know, this tRNA really scans that region for a codon that's really favorable, right? Because it really wants to be able to make the correct protein and not have to deal with having a truncated protein in that case. Another thing that affects the efficiency of translation in eukaryotes is mRNA secondary structure. So depending on where the secondary structure occurred, right, this RNA will fold back on itself. You can either have a positive or a negative effect depending on where it is, okay? So a hairpin just past the AUG can force the ribosome subunit to pause, and it will help to stimulate translation, right? So just past that AUG. If you have a hairpin or a stem loop structure between the cap, which the ribosome recognizes, and that initiation codon, then you will inhibit translation, right? Because the ribosome cannot get past that secondary structure to get to that AUG. And this is a negative effect on translation. And this is just an example of the experiments that they did to prove that, of course. This is, you're looking at an agarose gel, different constructs, different lanes, and you're looking at translation. And this is methionine that has been labeled, so you can see the band if it's been translated. For these different constructs, they engineered these constructs to be, you know, these 4 different versions, where you have construct 1, you have the 5' cap, 12 base pairs downstream of the 5' cap, you have this huge hairpin structure, right? For the rest of this gene. They also have between the cap and the secondary structure, 52 bases, right? So the AUG is here. You have a construct which there's no hairpin structure, this is AUG, so this is kind of your control. And then you have a hairpin structure, 71 base pairs downstream before this AUG, right? So you have these 4 different versions. When you do a translation assay, adding ribosomes with these messenger RNA, what you get is, for number 3, you're going to have translation, right? Because there's nothing inhibiting, there's no secondary structure, you get translation. That's what this band is showing. For number 2, you also get translation, right? And you're like, why do you get translation if the AUG is kind of trapped in this hairpin structure? So the ribosome will scan along. The ribosome is able to unwind some part of this DNA, right? So it will be able to unwind, and it's not a very large structure like here. It can unwind this RNA. I think I said DNA. It can unwind this RNA and access this AUG. So it will still go through and do translation, okay? In case of 1, you don't get any translation because this is 12 nucleotides. It's not even enough for the ribosome to fit there, right? So you cannot even get translation, right? If it's, you know, these are kind of the same size, but the spacing here is really narrow compared to this. So the ribosome can't even get there to even assemble there, much as to start doing anything. And this is just showing you when you combine 1 and 3, you can get stimulation because this doesn't prevent this from being translated. In number 4, which is what you expect, there's a huge hairpin structure even before the AUG. There's no way the ribosome can even overcome all of this, unwind all of this to get to the AUG, and it's not worth it, right? Here, the AUG is somewhat accessible when they unwind it. But here it's like 71 bases. You go through this really large hairpin structure, then you still have to go along to this AUG. That inhibits translation. You don't see it here. Okay, and then 3 and 4, again, if you add this to this, you can still translate this because this is not affecting it. Yeah, yes, question.
So regardless of where the hairpin structure is, as long as it has that AUG in it, then it should still be able to run through with the translation, as long as it's not before.
Here. Not necessarily, because in construct 1, you see you have it here. So if it's very close to the 5' cap, it doesn't work, right? So you need enough space for the ribosome to be able to be assembled in the mRNA. And if that AUG is in that hairpin or secondary structure, it can unwind it and overcome that and translate. Here it can, because the ribosome cannot assemble here.
Okay.
Here.
Thank you.
Mm-hmm. Okay, so I'll stop there simply because I'll stop there and continue on Thursday because I want to kind of do a little bit of some of these poll questions and give you guys room for asking additional questions. So we're all thinking translation, I know. But we just also talked about CRISPR. And I know it's kind of hard to kind of switch gears between the two, but I want you to be able to think about this because this is how the exam is, right? You have all these different concepts that's being thrown at you. So we went over the guide RNA extensively, talking about what is the role of the guide RNA. So yes, I think most of you are kind of on the right path, right? I mean, it ensures that, I mean, of all of these answers, right? The cell recognizes that DNA is damaged and tries to repair it. That's not the role of the guide RNA. That's the role of the different enzymes that's involved in DNA damage repair. Ensures that Cas9 cuts at the right position in the genome, right? That's definitely correct, right? You can say in one context, you can think of this as